Okay, I'm going to go ahead and get started. If you happen to be in intro bio last semester, hello again. I'm the one that did the research project thing in 150 and 151. If not, my name is Jana. I am a graduate student here. This is my second year. I am focused on biology education, and so I'm really interested in this, and so jump at the chance to lecture whenever given the opportunity. And so when Dr. Anderson asked me to fill in for what is 100% my favorite class, couldn't pass it up. So today we're going to start the animal diversity chapter. I personally think this one is pretty cool because it's like, I don't know, you know a lot about animals just from being alive, being a kid, reading books, those sorts of things. And this is going to have some new information about the breakdown of animals and the traits that they have. But I think it's really cool to see how we go from something like a sponge to like arthropods or vertebrates or things like that. Maybe. Okay. So to start off, animals are monophyletic and they are eukaryotes. They are multicellular, and the key thing about them that makes them different from plants and fungi is that they lack cell walls. Animals are also heterotrophic, so they have to obtain carbon from outside sources. They can't make it themselves. There are some animals, like coral, that have a symbiotic relationship with like things that are photosynthetic, but they're still taking in the products that something else made. And then most animals ingest food and then absorb it like in the digestive tract. So what humans do, you have to eat something, and then once it is broken down in your stomach, then you absorb the nutrients. But there are some animals that can just absorb it across their body surface. Like if you think about sponge and sponge-ish things that are just like floating in water, they can just take it in like as it passes across them. And this diversity of ways to obtain food is kind of what might have led to the diversity of animals that we see. Because if you think about animals versus plants, there's only so many ways that a plant can have photosynthetic tissue, present it to the sun, get enough light to make sugar from it. But there's a lot of ways that you can obtain food. You can be a spider that liquefies an insect and then takes it in, or you can be an anteater that's got this really long sticky tongue, so you can get into the tiniest little places to get ants and termites, or you can be a lion that's going to take down prey and eat really big things. And there's just a lot of ways that you can be an animal and be a successful animal. And then animals are also consumers, and that's because they are heterotrophic, so they have to take in food. So they are consumers in lots of ecosystems. And we care about animals and learning about them because like we encounter them a lot as humans. They impact our health with like transmittable diseases, and we use them for welfare. There's industries involving animals. We eat animals, obviously. There's just lots of ways that we encounter them. So we're going to start by looking at how we got to the animals. So we're starting here with this tree, which down here we have the Uniconta, which I'm assuming you guys have gone through these terms, maybe? Anybody? Yeah, okay. So down here in Uniconta, that's where we find our animals. And we're going to walk through how we get from this all the way through the choices. So if we look over here, this is our tree of the animals. Metazoa is another word for animals. It's synonymous with like Animalia. Metazoa is the same thing. And then we're going to go through the characteristics that like differentiate all these groups and that we picked up along the way. Most of these species or these different phyla you can see arose after the beginning of the Cambrian era, whatever the Cambrian is. And so this is where we have the Cambrian explosion, if you've heard that before, where we suddenly got all of the animal diversity. And most of these came after that. And so as we're going through this, we'll kind of walk through like the different traits that animals have and then circle back to talking about like specific lineages within it. And as we're going through, you want to focus on, like, you want to just kind of keep this tree in mind and think about the organization, why it's organized the way it is, what is more closely related to what, and then we'll talk about the synapomorphies that got picked up or lost along the way. But this tree is just, whenever I took this class, like it was constantly flipping back to reference like this kind of tree. Okay, so have you guys talked about the choanoflagellates yet? No? Okay. So the choanoflagellates are the outgroup to animals. So if we flip back to this tree, they would be like coming off up here above the tree. They're the outgroup to all of the metazoa. Choanoflagellates are these cool little cells that just kind of like float around in the water and take in food particles and stuff like that. But they can form this colonial structure. They can kind of congregate together to become something that has multiple pieces to it. And the similarity between choanoflagellates and choanocytes in sponges, like the name similarity is not an accident there. We see that these sponge feeding cells that like line the inside of the sponge structure look a lot like these choanoflagellates. And so it's kind of thought that sponges might have come from the aggregation of these choanoflagellate cells, and then they eventually evolved into just one like permanently colonized structure and became a sponge. Choanoflagellates have certain like genetic connections to animals, because you probably wouldn't look at that and think that it is anywhere even remotely close to an animal, but there are like protein similarities that choanoflagellates share with animals. And so like things for cell adhesion or cell signaling, which if you think about the like cell adhesion and cell signaling are things you need if you're going to be a multicellular organism. You need your cells to stick together and you need them to be able to talk to each other. So that's why these are kind of thought to be the precursor to the multicellular animals. So whenever we think about the evolution of animals, there are kind of four main things that happened. The broadest patterns of change that we saw were the origin and elaboration of tissues, the development of a more complicated nervous system, different symmetries evolved, and then this fluid-filled body cavity. So if we think about these, the origin and elaboration of tissues, like where do you get different tissues? What makes up a muscle? What makes up skin? What makes up nerves? Like all the different things have different tissue origins. And then the nervous system, it just becomes more elaborate, and we'll get into all of these on the next couple of slides too. And then symmetry, we start seeing things that are bilaterally symmetric, so have like a left and right side instead of just being a round blob that floats in the ocean sort of thing. And then we see cephalization, so the development of a head and rest of body. And then the evolution of a fluid-filled body cavity, which is kind of a complicated thing to think about and is one of those things you kind of have to just sit and chew on for a while, and then eventually it'll like click how all of these things are organized. We'll get more into that in a minute too. But if you think about like in your body cavity, you have the outside wall, and then you have like the inside wall that is like the outside part of your digestive tract, and then you have all of your organs and stuff in between digestive tract and the body wall. They're not like whenever you eat food, it doesn't go hang out with your liver. It stays in the digestive tract, which is separate from where all the organs are. But again, we'll come back to that in a second. Any questions up until this point? Yeah. Can you say that again? The cephalization kind of falls under both the nervous system and the symmetry stuff. I'll elaborate on that more once we get there, but it's kind of important for both of them. And we'll see that like the changes happen all kind of together. Like we talk about them as separate things, but like they all happen at once and then you get a bunch of animals for the most part. Okay, so to start breaking these down, the first thing we're going to talk about are the tissues and the developmental origins of them. So our three flavors of embryonic tissues are ectoderm, endoderm, and mesoderm. So if we break down those words a little bit, ectoderm, ecto means outside, derm means covering or lining or something like that. Endoderm means inner, and then covering or lining. And then meso is what's in the middle. Meso means middle, and so it's the middle lining. It's whenever we're talking about a lot of these different axes of diversification for animals, it's really helpful to like pick up on what the roots of the words were, because that tells you like they were named what they were for a reason. So pay attention to like the roots of words, just kind of in general in science classes, honestly. There's some scarier words down here. You don't need to know these words. Don't worry about those. The purpose of this figure is so you can see that like in development, the different tissues are present. And then like, for example, we know that the gut is made of endoderm because it's the most inside part. And so you can see here, like in development, the gut just kind of starts forming from within the endoderm. And then it's like lined by the yellow part, which is the endoderm. And then the red part would eventually form muscles. And then the blue part is the ectoderm, which would be like the outside skin. So then in terms of the tissues, so we have our three choices. There are some animals that have all three of the embryonic, they only have two of the embryonic tissues, and we call those diploblastic. Di means two. And then we have some that have all three of them, which are triploblastic. Tri means three. So if we look here, this is a cross section of like a hydra, I think, like the little jellyfish related thing that kind of looks like a little palm tree. It only has the endoderm layer and the ectoderm layer. That's it. And then over here, this is a worm. And in the cross section here, you can see that we have all three layers, the endoderm forming the digestive tract, the red mesoderm that is going to form this body cavity and then house the organs within it. And then we have the blue ectoderm, which is the outside covering of the worm. And this is the same like basic plan that everything that's triploblastic has. Like this is very simplified of what a cross section of you would look like. There's your digestive tract, there's your body cavity, where all your organs are, and then the outside covering is your skin. The interesting thing are the sponges. So if we flip back to our tree here, sponges come off on their own little group, and then we get to the eumetazoa. So the eumetazoa are the true animals. If you've heard the like prefix "u" before, you know that means true. And so these are the true animals, and everything from here on down has tissues. Sponges, on the other hand, don't have any tissues. So they're not triploblastic, they're not diploblastic, they don't have endoderm, mesoderm, or ectoderm. They just are some loosely organized cells together that are not organized enough to be actually considered tissues. And so that's what kind of causes them to be off up here on their own. Like they're animals, they're within Animalia, but they're like the sort of animals, and then we have the true animals starting at eumetazoa. And so one of the other key things to note about the tissues here is that mesoderm is where our organs come from. So these things that are diploblastic that don't have mesoderm don't have organs. And so those are things like cnidarians, so like the hydra and jellyfish, they don't have organs. And if I showed you a picture of jellyfish, you probably would initially respond with like, no, it doesn't really look like it has all that much in the way of organs anyways. And so that's kind of why the diploblastic versus triploblastic matters, is because that determines whether or not it can have organs, which obviously affects how complicated that organism can be. So the next part is going to be the elaboration of the nervous system. So our two different choices for nervous systems are nerve nets and a central nervous system. So this figure here, this is the hydra that I mentioned. It has what we call a nerve net. And so there is no like central control center for it. It's all just like responding to the environmental stimuli. So like something comes along and brushes one of these arms and it does whatever a hydra does, flicks it or something, but there's nothing like telling it to do that. It's just a nerve responding to a stimulus. Then we get to things that have a central nervous system, which have some kind of brain, or like ganglia is a brain term if you haven't heard it before. So that is like a really simplified version of a brain you can kind of think of. So this is like the end of an earthworm here. And so we wouldn't say it has a brain, but it has a collection of ganglia, which is brain-ish enough for this to be a central nervous system. And so this is where the cephalization thing kind of comes into play, that if you think about it, if you're an organism that is actively moving through the environment, so not a jellyfish that's just kind of like floating along, then you would want all of your organs that perceive things to kind of be at the front of you so that you have that going through the environment first. Your brain that does the controlling, you want to encounter the new stimuli first and then tell the rest of you what to do as like a second thought. And so that's where the cephalization comes into play, is that we can put our brain and eyes and ears and mouth and nose in one spot that's going to pick up the environment first, that we can then integrate with the brain to tell the rest of the body how to react to whatever situation we just encountered. And so this is how the cephalization and nervous system thing kind of come together. And so again, cephalization, you get the head or an anterior region. It's not always the most head-looking head. And then some kind of ganglia or brain. And then this will lead to the body symmetry, which we'll talk about on the next slide, and also is useful for things that have movements they're trying to coordinate to move through an environment. Any questions on any of that so far? Okay. So the next major thing that allows animal diversity is the body symmetry. So here are two different kinds of symmetry, are radial symmetry and bilateral symmetry. So radial are things like cnidarians, so jellyfish again, that are just kind of round, or like a sea anemone. It's just like a round thing. There's not a left and right side to it. And then these are usually things that are either attached or not actively moving. So like this is a top view of, again, the hydra thing. So they are just like stuck in one place and let the ocean bring food to them. Or you have something like a jellyfish that is not, like they can contract themselves to stay afloat to a certain extent, but they're mostly just relying on the ocean currents to carry them places. And then everything else comes with bilateral symmetry. And so that's everything that you think of when I say animal has a left and right side, and that kind of comes from having a head that it starts as your like defining point on an organism and then leads to left and right and that kind of thing. And these are usually unattached organisms that are like moving through an environment. I don't want to say by choice, but I guess kind of by choice, like coordinating their own movements, not reliant on the environment. Yeah. Bilateral. So they have, if you were to cut through an octopus, you would see like left and right sides of organs and stuff. We'll talk about the mollusks in this lecture at some point. And they're kind of an interesting thing because like you would never like crack open a clam and think that thing has a head, but like it technically does. So we'll get there and there'll be some pictures and stuff. Yeah, that's pretty much it for the symmetry. Okay, and so this is a little activity that Dr. Anderson wants us to do. So these are all pictures that he took on a trip to Olympic National Park, I believe, in Washington. And so here we have a banana slug, then we have starfish, and then back here is a sea anemone, and then this goat. So what kind of symmetry do each of these things have? Somebody tell me what kind of symmetry the slug has. Bilateral, right. And then what about the sea anemone? Radial, because that's in our cnidarian group with our jellyfish and stuff. So it's just this round thing that doesn't have like a head or anything. And then what about our billy goat friend? Bilateral, right. And then what about the starfish? So it's bilateral, believe it or not. Does anybody know the trick to this question? Yeah. It does. It does. That's on the right track. That's not the secret here, but that's on the right track. So starfish actually, as adults, have radial symmetry. So we would say that this thing has radial symmetry. But as larvae, they have a left and right side. They lose it as adults. But as larvae, they are bilaterally symmetric. So that puts them into the Bilateria group of the tree and is enough to get them in the club of all the bilaterally symmetric things. So there's your fun fact for the day, that starfish actually are bilaterally symmetric. Okay, so then next, this fun fluid-filled body cavity thing. So the cavity itself is called a coelom. That's how you say this word right here. And it's the compartment within the body that you can see like around, like if you're not in the digestive tract and you're not outside the body, you're in the in-between part. It's the cavity that has like, it allows for the circulation of oxygen and nutrients and helps to support the organs and allows them to, like the organs are mostly going to be like connected to the inside of the cavity. So if you've ever heard of like the peritoneum is like where all of your abdominal organs are, like that's the peritoneum is the peritoneal cavity is the cavity that would be the coelom. Like your coelom in your abdomen is the peritoneal cavity, if you've heard that before. That's another way to think about this. But it's just where internal organs are housed. Like we've already said, the diploblastic organisms don't have a coelom because the coelom comes from the mesoderm, and diploblastic animals don't have a mesoderm, and so they can't have a coelom, and therefore can't have organs and stuff too. And so our things that don't have a coelom because they don't have mesoderm are the cnidarians and ctenophores, and then the sponges, which don't have anything at all. And then our triploblastic organisms have a coelom, so they're like our little earthworm over here, where the organs and stuff are going to be within the body cavity, but separate from the digestive tract. They all have a coelom, unless it's lost secondarily. I don't know of any examples of that, but animals are weird, so I'm sure something loses it. And they're also going to be bilaterally symmetric. And so again, like they all these traits kind of pop up at the same time. And the coelom body cavity is one of the major traits of animal diversity because, like I said, if you don't have mesoderm, you don't have a coelom, you don't have organs. And so once we started having mesoderm and coeloms, then we could have organs, and then that's when diversity really could take off, and the way that things looked started really diversifying, because I'm sure there's a lot of like sponges and cnidarians and ctenophores, but they all kind of look the same. They're oceany things, but then once you get organs and you get like the animal, when I say animal diversity, you're thinking of things that came after the coelom organs thing started. Okay, so now we're just going to kind of like summarize some of the major takeaways of what we've just talked about so far. So if we go back to our tree here, remember that choanoflagellates are the closest living living relative. So they would be the outgroup up here at the top of this tree. That's where we would put our choanoflagellates. And then we start here with metazoa. It's the same thing as saying animalia. And remember that all of these terms that end in like the A are things that are monophyletic groups. So like you would say, the fact that you can say animalia means that it's monophyletic, as opposed to just having to put like an S on something and it's just a group we talk about that isn't monophyletic. But everything on here, all these terms are monophyletic groupings. And then our first thing within the metazoa are the sponges, the porifera. They are the sister group to all the other animals. They're the outgroup to the eumetazoa. There's multiple ways that you can think about the breakdown of this. And then our next breakdown is the bilateria here. And so this is where we pick up pretty much all of the methods of diversification that we've talked about so far. So that's where we start having bilateral symmetry, triploblastic tissues. So we have mesoderm, so we have a coelom, so we have organs, we're cephalized, like all of that stuff comes here at the beginning of the bilateria. The closest relatives to the bilateria are going to be the cnidarians and ctenophores. Those are like the cnidaria would be the outgroup, sister group, whatever you want to call it, to the bilateria. And so we know that first we had to have the endoderm and ectoderm because that's what our more ancient things have. And then the mesoderm had to come after that because that's what all of our more derived things have. So we know that the ectoderm and endoderm had to come first. And then since organs come from mesoderm, we know that the mesoderm had to show up before the coelom that can house the organs before the organs. So try to think about, like me just rattling all of this off to you is you're not gonna just walk away from this having all this memorized, understanding all these relationships, but this is kind of how you should be looking at the tree. Like you should be able to just like point to a spot on it and tell me what came before it, what the things before it looked like, and what the things that came after it looked like, what traits were picked up or lost or whatever. And like we said, most of the bilaterian lineages evolved during or after the Cambrian explosion. So like we already had these sorts of things, but all of these things pretty much came after the Cambrian explosion. Yeah, and so then we're going to start focusing on the bilateria for the rest of this lecture and start actually going through some of these groups over here. Within the bilateria, there are three major lineages. So the ecdysozoans are these things down here. So they include like bugs and stuff. And then the lophotrochozoans are all of these things right here. So that's where we have like mollusks and earthworms and things like that. And then the deuterostomes are the things up here, which are echinoderms, starfish, and then chordates, vertebrates, most of what you think of when you hear animal. The tricky thing here that you may or may not actually encounter is this word protostome. Actually, that's spelled wrong. That should be protostome. But anyways, it's a word that when I took this class in 2018, we called the ecdysozoans and lophotrochozoans the protostomes. So we had the deuterostomes and the protostomes. However, in the last couple of years, even like the book that I had, I think was 2017. And so between the 2017 book and the 2020 book that you guys use, scientists have decided that there actually isn't anything that like the synapomorphies that they have for protostomes actually don't apply anymore. And so now we've kind of dropped that term. And so we have just these three. So notice that like on this tree, the lophotrochozoans and ecdysozoans are not like the same level as deuterostomes are. Like these are not the same rank of term. We would have protostomes right here, but we've since dropped the term because whatever we thought applied doesn't apply anymore. Somebody found something that contradicts it. So this word is, you might still see it every once in a while, but it's like technically not accurate at this point. And I looked in the book and it only mentions it like one time way later on in the book. Like I don't think this chapter actually mentions it, but if you see it, this is the secret to protostomes. And then lastly, we have if we focus on the chordata, which are all the things that have like some kind of nerve cord, spinal cord, that kind of thing. And then within chordata, we have the vertebrates, so the spine things, backbone things. These are a monophyletic group. And so this includes everything from like fish to reptiles to birds to mammals. Like most of your animals that we think about are in the chordates and vertebrates. So the vertebrates are all within the chordates. So they're a monophyletic group. Would invertebrates be monophyletic or paraphyletic based off of this tree? It would be, because of these fun little things called echinoderms that are more closely related to the vertebrates than they are to all the other invertebrates. Like the starfish is more closely related to you than the octopus is, which to me is kind of weird to think about, because like there are, not that there's, these feel less complex, I guess, but like I would definitely picture an octopus that has like eyes and you can tell is thinking about things as being more closely related to me than a starfish, but that's not the case. Any questions about the major diversification takeaways? No? Okay. Oh, so this tree, it's like almost the same as this one. It's missing a couple of the groups, so it's not like 100% accurate, and it's still got the protostomes word on here, but it's got some of the like synapomorphies listed on here. Like see all of these things happen together. And so this is just a good place to start for like remembering where the synapomorphies happened on the tree. Okay. Now we'll actually get to talking about what some of the groups are. So we're going to start with the lophotrochozoans and ecdysozoans. So these guys right here. These are like the biggest groups of animals. Most of the animals are contained within this. If you look at this figure, which still has the wrong word on it, but we can ignore it for the purposes of this, like all of these things that are in color are within the lophotrochozoans and ecdysozoans, and then just these things down here are the deuterostomes. Each of these respectively is a monophyletic group. All the Lophotrochozoans are most closely related to the other Lophotrochozoans. All the ecdysozoans are most closely related to the other ecdysozoans. And if we look at this, most of these two groups are comprised of insects, which I'm sure that you are aware that there's lots of bugs, lots of different flavors of bugs in the world. So it kind of makes sense. It's kind of intuitive. And these things are all passed. They're all within the bilateria. So we know that they are triploblastic, bilaterally symmetric, and coelomates are, that's the term for the things that have a coelom. So they have a coelom, they have organs, that kind of thing. So then looking at just the Lophotrochozoans. So here, this name, this really long name, comes from the lophophore term and the trochophore term. And then we just kind of mushed them together and put them into one group because like genetically, they keep shaking out as all being the most closely related to each other. The lophophore is this feeding structure here, which like they can put out these little really looking cilia covered things, like little hairy things, and then the water moves across them, the food particles get trapped and then brought into the organism through this little mouth and gut and then back out to the anus. And you don't need to know the details of how this works, you just need to know that lophophore is a feeding structure. And then trochophores, that's a word for a type of larvae. So like these two things, lophophore and trochophore, are not the same thing. It's kind of apples and oranges. They just keep being genetically related to each other. Trochophores, it's the word for this larvae. This would be called like a trochophore larva. And these are what the babies of like mollusks or something would look like. They kind of float through the ocean and then eventually, like if this is, I don't know exactly what this is, but we'll say it's like a clam baby. So this thing will kind of like float through the water and then find a place to settle and then become like a clam or scallop or whatever. So it's just the term for the larvae. And the thing is, neither one of these are synapomorphies. Not all of the Lophotrochozoans have both of these, or I guess, well, yeah, I guess none of them, not all of them have both of them. And so they can't just be like synapomorphies for the whole like group here. If it was, then we would put them right here, but that's not the case. Some of them, most of them have them, some of them have one of them, but they're just like traits of the group. Then the still on this. We'll go through some examples of it. So the two major lineages of the Lophotrochozoans that you will recognize and should remember are the annelids and the mollusks. So annelids are segmented worms. So that's things like the earthworms or like leeches. And then these are oceany things. So like a leech is a segmented worm. And then mollusks we've talked about. Oh, actually, let me make a point here. So even though something like an earthworm is like round, it's not radially symmetric. Like it's still, if you were to cut this thing down the middle, it still has a left and right side, still has a head end and a tail end. So don't let just like round animal trip you up. Okay, and so then mollusks are our other like main part of the Lophotrochozoans. So you would never like look at the inside of a clamshell and think that thing has all of these things we've talked about: symmetry, a head, complicated nervous system. Never would you think that, but they do have a very specific breakdown of the body. So we have the body encased in this structure called a mantle, which is the thing that's like unique to mollusks. And that's what ends up secreting the shell in some of them. Not all mollusks have shells, but if they do, it comes from the mantle. And Then you have like the foot down here, which allows it to move, which I got to experience that for the first time this summer, like being on the beach and having like a live something in my hand and feeling the foot like suction to me. It was very cool. Highly recommend if you ever get the chance to pick one up. But here are some examples of them. So we have things like snails that have a shell, or things like slugs that don't have a shell. And then we have our like clams and bivalves and scallops and mussels and those sorts of things. And then down here is the cuttlefish. And so it has like sort of a shell to it, but not like a complete one, like a snail. And then like a squid or something has one bone thing in the middle of it that's kind of functioning as a shell, because it doesn't have like a skeleton backbone or anything, but it does have like a bone in the middle. And then we have octopi, which are just one of the coolest things. They're so smart. If you've never like, I don't know, looked up anything about octopi intelligence, would highly recommend it. There's a video that exists of an octopus that's inside of a jar, and it figures out how to unscrew the lid and get out from inside the jar. And it's an octopus. And they can learn by watching. So you can put one in a tank next to one that figures out how to do a jar, and then you can put that one in a jar and it knows how to do it. And it's an octopus. It's just the wildest thing to me. They're so cool. Highly recommend doing some research on them. Okay, so moving on to the ecdysozoans, our other flavor of lineages of the bilateral things. So these do have a nice convenient synapomorphy. They all grow by molting, which is ecdysis is the sciencey word for the process of molting. And that's where they get their name from. And this is a synapomorphy that they all do. And so most of our named species are within the arthropods. So insects, spiders, zooplankton, crabs, crayfish, all of those sorts of things. They all have segmented bodies organized into tagmata. This word is just like the term for the segments. So like head, thorax, abdomen, those insect words you've heard before. That's what this line means, is it's organized into those different segments. Jointed limbs. And they have an exoskeleton that is made of chitin. And the muscles are able to pull on that exoskeleton to get the organism to move, which you'll probably hit on again once you get to the muscles chapter later on in the semester. But you'll talk again about how muscles move a skeleton and stuff like that. And then there's some more examples down here. So like spiders, and we got dragonfly. This is a little zooplankton thing. I think it's called Daphnia. I think is the word for it. But that's what that is. And then lobsters and lovely cicada over here, which we're all very familiar with. But yeah, ecdysozoans. And then lastly, there is the coolest ecdysozoan, tardigrades, which is not tardigrade, like tardigrada is a phylum. So if we go all the way back to one of the trees, like we could put tardigrada as another little branch coming off down here, but the book neglects to do so, which Dr. Anderson was not all that pleased with, because they actually have found some of these, his lab and his students have found some of these in the ponds on campus. So like the one that's behind Evergreen has water bears in it, which I had no idea until I was talking to him about this lecture, that like, wait a second, they're not just like a faraway thing, like we have tardigrades here. But anyway, so these are, it's a phyla within the ecdysozoan. So that means that they grow by molting, have that synapomorphy. But yeah, they're just one of the coolest things ever. That's all that I have for you, though, I think. Last slide. Thank you for your attention, even though I'm not your professor. Yeah, question? I don't think he's specifically studying them. That was just like a happy accident to find that we have them, but maybe. It could be. Yeah. Parent, I mean, they might be. I just know we have them here. That was the takeaway. I have no idea. Yeah.